I am Garret Kingman. I'm a NASA postdoctoral program fellow in the Rothschild Lab at Ames Research Center. My background is in evolutionary genetics, and I'm very excited to talk to you today about my work applying these ideas and techniques from evolutionary genetics towards understanding the limits of and opportunities for life. So the big question I'm interested in, and I suspect all of you are as well, is what are the opportunities? Answer in theory. We simply take the set of environmental conditions that are permissive for life, and we intersect them with the environments present on extraterrestrial planets, moons, and other bodies. And the overlap represents the opportunities for life. But in practice, it's not that simple. There's a lot that we don't know about the physical environments of planets and moons, even within our own solar system. And there's also a lot we don't know about the environmental conditions that are permissive for life. NASA, ESA, and many other organizations deservedly place a large emphasis on searching for life through a variety of missions to characterize planetary environments, and work constraining the other side of the equation, the environmental conditions permissive to life, largely consist of characterizing the highest temperature, most radiation, pH extremes, and so on of what we know life on Earth can do. And this kind of work studying known life is extremely valuable, but it does suffer from one key bias, which is that our knowledge of the limits of life is fundamentally limited by all known life originating in one place, the Earth. And this represents a large problem because we cannot readily disentangle the actual biochemical limits of life from the limited selection pressures that are found on Earth. So when we look beyond Earth to Mars or Venus or Europa or Enceladus and many other places in our solar system, there are all kinds of environments and environmental conditions that are just not found on Earth, that terrestrial life has never had to adapt to. And so when we try and ask the question of could life survive here, we're asking it of organisms that we have available, terrestrial life, that's never had to adapt to anything like this before. And importantly, these extraterrestrial environments, as we heard already from Piotr, they contain not just one, but multiple environmental challenges. You can think about each one, such as we heard from him about temperature and pressure affecting membranes, but you can also imagine salinity, pH, radiation, desiccation, salts, metals, pressure, gravity. The list can go on and on and on. And you can visualize all of these as kind of forming a highly multidimensional space. And some conditions within this multidimensional niche space are compatible with life, and others are not. And we don't know which is which. We know some that definitely are compatible with life, but there's a huge amount of other conditions that we just don't know for sure. And as life on Earth has evolved in the conditions found on Earth, the niche space of known life very closely mirrors the range of conditions that are found on Earth. And while Earth contains a huge diversity of environmental conditions, it is nowhere near the range that are out there in the solar system and beyond that could potentially be harboring life. And one last point I want to make here is that before moving on to my actual work, is that while I've illustrated things here as a simple sphere, not all of these combinations of environmental challenges are equally tolerable. We'll see a few examples throughout this talk of how adapting to one particular challenge may either facilitate or impede adapting to a different environmental challenge. So what are the opportunities for life in the universe, and how do we approach this question? My goal is to try and decouple our knowledge of life's capabilities and limits from the limited selection pressures on Earth, and to do this by trying to expand the known limits of life. So today I'm going to be telling you about 3 approaches I've been taking to this problem, starting with the first one, using a technique called adaptive laboratory evolution. This is where you apply a selective pressure to a laboratory culture, such as adding perchlorate and increasing amounts to the growth media, and allowing your cultures to grow for hundreds or thousands of generations. Over time, genetic mutations will arise. Some of them will help an organism adapt to the environmental stress, and these will naturally increase in frequency over time, hence the name adaptive laboratory evolution. So these are just a few of the conditions I've been playing with: pH, high and low, salts, including both sodium chloride and perchlorates, which are much more common on Mars, but very rare on Earth, and UV exposure. And I've been doing this with a couple test organisms, namely E. coli, Bacillus subtilis, and Deinococcus radiodurans. And I'm not going to tell you, I'm not going to go in detail about the specific methodology here for everything, but I'm going to talk about some results in Deinococcus radiodurans. And the key question here is, can polyextremophiles, which Deinococcus radiodurans definitely is, and I'll talk more about it in just a moment, but can polyextremophiles that are already really robust at dealing with multiple challenging environments, can they become even more extreme? You could imagine if you were mapping out the niche space initially, for example, when you try temperature and pH or temperature and UV, they could grow. And with adaptive laboratory evolution, they could be able to tolerate more than they could originally in their native form. And they don't necessarily grow in a uniform manner. It could be that some combinations of environmental conditions have much greater resistance to expanding their niche space. So Deinococcus radiodurans is a polyextremophile I'll be talking about frequently over the next 10 minutes or so. As the name suggests, it is highly resistant to radiation. In fact, it was first discovered after a tin of meat was exposed to radiation to sterilize it, and it was thought to kill all known forms of life. But the meat subsequently spoiled, and people trying to figure out why ended up discovering Deinococcus radiodurans. It's actually been found in quite a few natural environments since then, including in lava tubes. And it's resistant not only to radiation, but also to desiccation or drying out, to cold, to vacuum, and to acid. But again, the question remains, can even so consummate a survivor as Deinococcus radiodurans become yet more robust and grow under still harsher environmental conditions? Because after all, like all terrestrial extremophiles, it is optimized for the environmental challenge it faces, not to tell us everything we may want to know about the biochemical limits of life. So here I'm showing you the results graph after many hundreds of generations of adapting Deinococcus radiodurans to grow in alkaline conditions. Here it is at pH 10. So here we have time on the x-axis and essentially cell count on the y-axis. And you can see that the evolved strain, in red, grows robustly at pH 10, while the original strain doesn't grow at all. This suggests that, yes, alkaline conditions certainly are something that Deinococcus radiodurans can adapt to. We can do this with other environmental stressors as well. So here I'm showing you a similar experiment, but for perchlorate, again, a salt that's very rare on Earth, but very common on Mars. And it can evolve a significantly increased performance and ability to grow in sodium perchlorate in its growth media. Interestingly, if you take a strain of radiodurans that we evolved to tolerate salt, as in sodium chloride, and then you ask it to grow in perchlorate, it lags initially, but ultimately grows very well, just as well as the perchlorate evolved strains, suggesting substantial overlap of these response mechanisms. Similarly, strains that were evolved with regular UV exposure have improved growth in these high perchlorate conditions, which makes sense because UV and perchlorate can both cause oxidative cellular stresses. In contrast, the alkaline evolved strain that we talked about just a moment ago actually grows much, much worse in the presence of perchlorate. And that's not just worse than the other evolved strains, but worse than the original wild type strain, kind of in this white and light gray. And this suggests that there is a fundamental incompatibility between the adaptive mechanisms for these two different types of stress. And so that when we think about adapting to environmental conditions, this really just illustrates that you can't consider them in isolation. You can adapt to alkaline, you can adapt to perchlorate. Adapting to them both together, it's different. So that's kind of the highlight of this first section of the talk. Even polyextremophiles can become more extreme. When we study them as we collect them on Earth, it doesn't mean that they're at their limits. It means they're at their limits for what they've had to do naturally, which is not the same as what they're capable of. That said, It can be difficult to trace back from these phenotypic results to understand the genetic changes. And sometimes a genetic solution to an environmental stress may exist, but be hard to access, which means that evolution can proceed very slowly or not at all, even if it is possible. And so the second part of the talk is about use of functional metagenomics to learn from extremophiles. And we can do this by providing them with genetic tools that can increase their ability to adapt to these new environmental challenges. Just because we're interested in novel environmental conditions or ones that don't exist on Earth doesn't mean that the challenges themselves are completely novel. Rather, it's a matter of degree and combination of these environmental stresses. And many useful genetic tools that are at least partial solutions to these problems do already exist. And so it doesn't necessarily make sense in an experimental laboratory evolution experiment to require that these tools be re-evolved. And one way that we can sidestep this is by introducing foreign DNA to give our test organisms novel capabilities. And this may sound exotic, but it's actually something that is done all the time. Many of you are, I'm sure, have done molecular cloning before, where you add a gene that provides antibiotic resistance, and then you can use that almost incidentally to help you clone your DNA constructs that you're trying to make for various experiments. We can do this exact same idea on a much larger scale by taking source extremophiles, extracting their nucleic acids, making a highly diverse genetic library, packaging it in a plasmid using lambda-phage, and then introducing the entire library comprising millions and millions of different genetic sequences into E. coli. And then we can screen them through adaptive laboratory evolution. And the few constructs that contain sequences that provide adaptive benefits will increase in frequency over time. And so through that manner, we can identify the tools used by these extremophiles without ever needing to culture them in the lab and at much higher throughput than is otherwise possible. Through this, we have been able to increase UV resistance by over 1,500-fold in our test E. coli. It identifies many different forms of the relatively well-known gene Garret A. And actually, every time I should speed up just a little bit, but the details of how you do this do matter. It's very strongly dependent on how large your copy size is. It turns out it's not just enough to have the relevant gene by itself. You also need to have the right genomic context, so it's expressed at the proper level, and the plasmid that you're cloning this all in be at the proper copy number, or it will yield unsatisfactory results. So the takeaway from this section is that it is very possible to raid nature's genetic toolbox to screen for useful genetic elements in a high throughput manner, and that this enables learning from a huge diversity of extremophiles without needing to culture all of them in a laboratory, which enables you to search through many more of them than is otherwise possible. And then just briefly, I want to touch on the use of motivated synthetic biology to do the same purpose. And the idea here is that we're fortunate enough to live in an age where there are huge numbers of data sets available to us, such as RNA-seq expression, a lot of whole genome loss of function screening, and we can use these to try and understand how we can increase tolerance. The caveat, though, is that these data sets, while present and extremely useful for many things, tell us they don't tell us exactly what we're looking for, and it's very possible to get to interpret them as telling us the same thing that we're interested in, when actually they're telling us something related but also different. So if you take genes that are, for example, upregulated in response to DNA damage, and if you intersect them with genes that are necessary for survival after DNA damage, there's actually extremely little overlap. And there are a couple ideas I have for why this is, but the long and short of it is that the modern abundance of data that's very useful doesn't necessarily make it easier to correctly identify which genes will increase fitness in a given environment. And so these types of data sets are all useful but give different answers. And so just remember, when you're thinking about data sets that are out there and how they can inform your experiments, it's important to design as closely as possible to your end goals. But we are in the process of synthesizing and testing some of these candidates anyway. So with that, I just want to conclude and restate a few important points. The limits of known life reflect the environments that life has evolved in, not necessarily the limits of life itself. The multi-parameter nature of life matters in considering possible extraterrestrial environments and in how adapting to one environmental challenge may either facilitate or impede adaptation to another environment. There's, of course, a huge amount still to do. The next step that I'm working on now is going from individual genetic tools, such as some of the ones I've told you about today and that many others have identified. We need to be able to combine them all into a toolbox where they can all work together to yield results better than any single one of them, because it's exceptionally unlikely that any single genetic tool, no matter how powerful, will be able to solve the immense challenges of Mars, Venus, Europa, or various other worlds in or out of our solar system. And so if there's one idea that I hope you all take away from this talk today, is that as we are all working to understand the possibilities for life in space, a fundamentally necessary step is not just to learn about planetary environments and to learn about terrestrial life, but also to break through the ignorance that we all have to deal with that's imposed by only studying life found on Earth. And by working to expand the known limits of life, we can significantly increase the known permissive conditions and thus opportunities for life. And in doing so, we just may find that there are more opportunities than we realize as we come to better understand what's possible. Thank you.